Hello everyone, my name is Claire McLean and I'm a marketing associate here at Omega Bio-tek. It is my pleasure to be moderating our webinar, Removing the Barriers of Automating Plasmid DNA Extractions. We'll be joined today by two of our internal experts, Dr. Kiran Durvasula, one of our product managers, and Jeff Roeder, our field applications manager. On the agenda for today, we'll start off with a brief introduction to Omega Bio-tek before jumping into the topic at hand, plasmid DNA and its applications, as well as diving into some of the automation capabilities for these workflows. Finally, we'll end this presentation by answering some of your questions, so please leave them below. And with that, I'll hand it over to Dr. Kiran Durvasula for a brief introduction to Omega Bio-tek.
Thank you, Claire, for the introduction. Hello, everybody. My name is Kiran Durvasula, and I'm a product manager at Omega Bio-tek. Omega Bio-tek is based out of Atlanta, Georgia. We are a 26-year-old company with our expertise in providing innovative DNA and RNA purification solutions. We boast a portfolio of more than 400 plus products encompassing different extraction technologies, including magnetic beads, silica columns, silica filter plates, as well as salt-out-based technologies. These methods are available as different kits and configurations covering a wide range of sample types. We are an ISO 9001, 2015 certified, as well as ISO 13485 certified, and our presence is felt around the globe through more than 52 distribution partners worldwide. At Omega Bio-tek, customer success is our top priority. Every project, regardless of scope or size, matters. We believe in delivering nucleic acid solutions that fit our customers' own unique needs. We understand the challenges of extracting high-quality DNA and RNA, and we believe in empowering our customers by making high-quality, automated products more accessible as well as affordable. We focus on collaboration, innovation, and integration, and we provide flexible purification solutions, both manual as well as automated workflows with protocols designed with our customers' needs in mind. With that, let's pivot to the main topic of discussion today, that is providing strategies for automating plasmid purification. Before that, I would like to present a brief introduction to plasmids and the applications that plasmids are increasingly being used in. Plasmids are small, circular pieces of DNA that are found in bacteria. Plasmids have their own origin of replication, so that they can replicate independently of the host genome and can produce several copies of the inserted gene. The important characteristic of plasmid is that the presence of the plasmid often is beneficial to the host cell, whereas the absence of the plasmid does not affect the functionality of the cell. For example, if plasmid has an antibiotic gene, it confers antibiotic resistance to the host cell. The other important characteristic of plasmid is that depending on the number of copies that the plasmid is found in the cell, it's often referred to as high copy number, as low copy number. Usually the small-sized plasmids are present in high numbers, that is the high copy numbers, and large plasmids, which are large size, are present in low copy numbers. Why are plasmids important? Plasmids serve as vehicles for communication of genetic information between cells. Hence, they can be used to introduce, manipulate, as well as delete certain genes from the host cell. These plasmids can be used to encode different genes and are able to transfer foreign DNA into organisms. As you can imagine, with this ability, the reach of plasmids is immense, and the applications that they can be used in is multitude. Some of the applications I've listed on the slide, they include some of the basic applications of sequencing, PCR, transfection, and protein expression, to more advanced applications of gene editing, gene therapy, to providing vaccine development and vaccine therapeutics. So purifying plasmids is the first crucial step to be able to use these plasmids in different applications. So the yield and quality of the plasmid is actually dependent on several factors, It's dependent on the host strain that harbors the plasmid. It's dependent on the culture volume that it's grown in, the culture media. It can be a high-density culture media like TB, or a more, basic culture media like LB. It's also dependent on the copy number that is present in the host cell, and also on the size of the insert. An important tidbit is the high copy number plasmids, which have a large insert, can actually act as a low copy number plasmid. So what this tells us is each of the system is pretty complex. It's a complex biological system, and each of the situations is heterogeneous. So essentially, you're dealing with a unique situation every time. So that makes plasmid purification a pretty complex process. So hopefully, by the end of the seminar, by the end of this webinar, we will equip you with more information on how to automate and how to deal with this heterogeneity in the plasmid purification technology. The different metrics that the plasmids are often measured are quantification, quality, as well as purity grades. I would like to go into each of these in a bit of a depth so that, while we talk about all these in the subsequent slides, you're all more aware. Quantification is basically to equip the researcher about the concentration of plasmid that is purified. And one would use nanodrop or absorbance-based systems for quantifying plasmid DNA. You would get information on concentration of plasmid as well as the purity ratios by looking at the A260, A280, as well as the A230 ratios. The quality of the plasmid can be ascertained by running the purified plasmid on a gel. Plasmid is actually found in different conformations. On the right side of the slide here, you can see the different conformations that the plasmid is found in. It can be nicked, linear, supercoiled, as well as circular, single-stranded. The desired plasmid conformation is the supercoiled. And an important point to note is that the supercoiled actually migrates faster than the predicted size. Also, by running the plasmid on the gel, one can get information about gDNA contamination or RNA contamination if it's present in the purified plasmid. Purity grade is another important metric of the purified plasmid. It is based on the endotoxin levels that are present in the purified plasmid. Endotoxins are lipopolysaccharides that are found in the cell wall of the gram-negative bacteria. And these often confirm immune response to the host cell and are often detrimental and can have consequences in different applications. So depending on the endotoxin levels that are found in the plasmids, there are 3 different purity grades. The first one is the endo-free, which is the advanced transfection grade, with endotoxins levels less than 0.1 EU per microgram. The mid-range is the low endo, which is, the lab grade transfection level. And the level of endotoxins for this low endo level is 0.1 to 1 EU per microgram. And the third is the molecular grade with high endotoxin levels, which are greater than 10 EU per microgram. So the purity grades of the plasmid often have a bearing on the kind of downstream application the purified plasmid can be used in. So this slide tabulates different downstream applications and the purity grade requirement for each of those downstream applications. As you can note here, the applications of vaccine development and research, sensitive cell eukaryotic transfections, gene therapy, and any kind of therapeutics which involves plasmid would need endotoxin-free purified plasmid. The transfection purity grade, for cloning and standard transfection, the endotoxin levels from 0.1 to 1 EU microgram are often required. And the basic research level, which involves applications of transformation, sequencing, PCR, one could get away with this molecular grade purified plasmid. Now that we have information about the plasma purity grades, I would like to shine light on the typical plasmid amount and scale that is needed for each of these downstream applications. For applications of protein expression, cloning, PCR, and transfection, the plasmid amount that is needed often ranges in the microgram levels. As we move on to the biopharma applications of gene editing, gene therapy, immunotherapy, the requirement is more in the milligram levels. And the next level is, the plasmids that are needed for vaccine production, vaccine therapeutics, enzyme production, and other large-scale plasmid productions would need plasmids that can range several milligrams. So essentially, there's no one solution that can fit all of these. And depending on the input volume and the amount of plasmid that is needed, we would need different solutions to be able to tackle those different problems. To answer this market need, Omega Biotech has recently launched 2 automation-friendly plasmid purification kits. One is the MagBind Endo-free Plasmid Mini Kit. This kit is designed to purify plasmids from up to 1.5 mil bacterial culture in LB or TB or other suitable growth medium, and is capable of extracting up to 20 microgram from high cell density culture medium. The second kit is the MagBind Endo-free Plasmid Mini Kit. This kit is designed to extract plasmid from up to 50 mil bacterial cultures in LB, TB, or other suitable growth medium. And it can extract up to 200 microgram from high cell density culture medium. Both these kits are fully automatable in the sense we use magnetic beads to clear the bacterial cell lysate, avoiding the manual centrifugation step. Both the kits result in endotoxin-free plasmid DNA. So essentially, they can be used in all the applications that I've shown in the previous slide. And they're sensitive transfection-quality plasmid DNA, and the endotoxin levels are less than 0.1 EU per microgram. And both the kits are versatile. What I mean by that is they're able to process different culture media, LB, TB, we've tested 2XYD, and they can handle different plasmid types, be it low copy number, high copy number, large size plasmid, etc. On this slide, I would like to provide a brief protocol overview followed by the kits. The kits follow alkaline lysis methodology. This is a molecular biology technique which differentially purifies plasmid DNA without gDNA contamination. And it has 4 basic steps. 1, lysis, where the bacterial cells are lysed under alkaline conditions. 2, denaturization, where the proteins and DNA are denatured into single-stranded. And third step is neutralization, where the alkaline conditions are adjusted, and the plasmid DNA being smaller re-anneals back. The fourth step is the removal of cell debris, where the genomic DNA and proteins are precipitated out and plasmid DNA is present in the lysate. Here's where the innovation from Omega Biotech comes in. Typically, the removal of cell debris is facilitated either by centrifugation or by using lysate clearance syringes. In our kits, we are providing magnetic beads to do the cell debris removal through magnetization. And what it does is this makes the entire protocol fully automatable and provides customers with more turnkey solutions for plasmid DNA purifications, which is hitherto not available, especially for higher culture volumes, sorry. So once the precipitate is removed, either by lysate clearance beads, centrifugation, or syringe, the rest of the steps follow a very routine bind, wash, elute protocol, where the plasmid DNA that is found in the supernatant is bound to a second set of magnetic beads, and it's washed several times to get rid of the contaminants, and it's eluted in endo-free water. And at the end of the protocol, you're left with the pure plasmid with endotoxin levels less than 0.1 EU per microgram and ready to be used in a lot of downstream applications. With that, I would like to introduce my colleague, Jeff Roeder, for discussing further on strategies for automating plasmid purification.
Thank you, Karen. My name is Jeff Roeder, and I'm the Field Applications Manager here at Omega Bio-tek. And today I'm going to discuss some of the different challenges or barriers that are associated with automating plasmid purifications, as well as some of the new workflows that we've developed for plasmid preps that utilize our new MagBind endotoxin-free extraction kits. So going into the challenges, and just to reiterate a lot of what Karen already mentioned, first off, one of the biggest challenges is just the variation among starting samples. There's many factors that can actually affect how your samples behave during the extraction process. For example, the growth conditions, the media that was used, the host cells, the plasmid copy number, even the size of the plasmid. All of these can affect your samples during the extraction process. Therefore, it's difficult to have a one-size-fits-all automation method that is robust enough to handle a wide variety of sample types. Another huge challenge of automating plasmid preps, and again, as Karen mentioned earlier, is the lysate clearance step. So there's a few different options on how to perform this step. Traditionally, either a centrifugation or a filter step is involved, and these are really both not automation-friendly options. But through the use of our magnetic beads, we've been able to overcome this challenge, and we've been able to develop some fully automated protocols, which we'll discuss more later on. Now, for higher scale plasmid preps, so for like MIDI or maxi preps, automating these protocols are very, very difficult simply due to the volumes of reagents that are required. And up until now, there's really been limitations with the hardware, the automated systems, and plasticware that can handle the larger volumes required for these protocols. And there's also been a lack of like scalable extraction chemistry or technologies. So for most techniques that are used for MIDI or maxi preps, they involve some sort of column-based extraction kit, and these are just not designed for automation. And the current options that are out there that can handle columns, these are very time consuming, and they can only process a few samples at a time. So there's a big issue there with scalability and throughput. So basically, through the use of our magnetic beads and with some of the advances in robotics and hardware, we have been able to overcome some of these challenges, and we've been able to develop some new automated workflows. Again, those higher scale plasmid purifications have traditionally been more difficult to automate. So I'm going to highlight some of our new solutions for those first. So the first workflow that I'd like to highlight uses our new MagBind Endo-Free MIDI kit on our MagBinder Fit 24 instrument. This instrument is a magnetic bead processor, meaning that it just moves magnetic beads around. It cannot pipette or transfer liquid. Therefore, this is a semi-automated workflow. The upfront steps, so those alkaline lysis, the neutralization, the lysate clearance steps, those would have to be performed manually. And then the DNA bind, wash, elute steps would be automated on the MagBinder. So it uses a specialized reagent cartridge shown here, with the first well having a large capacity that can hold up to around 12 ml of volume. So you would load your sample lysate, your binding buffer, and your magnetic beads into this well, and the instrument would mix the samples, and then it would transfer the magnetic beads to the adjacent wells that would contain the wash buffers and the elution buffer. So this workflow can process 24 samples per run. The manual upfront steps take approximately 30 minutes. And this can vary slightly from user to user, and it also depends on which lysate clearance method is used. And the automated processing time is roughly 70 minutes. So this would be a great option for low to mid throughput users, or for someone who needs to process anywhere from 10 to 48 samples per day. So briefly, just to review some comparison data, here we processed 5 ml cultures using this workflow. And basically the main takeaway here is that we're achieving very high DNA yields right in the expected range. And we're also seeing very low endotoxin levels, which would qualify under that endo-free grade. And this is contributing to a higher transfection efficiency, which is shown here as well. And here we process samples from 25 ml LB cultures, 50 ml LB cultures, and then 10 ml TB or terrific broth medium. And we basically use the 3 different lysate clearing methods for each, and the yields and the purity are comparable between all the different methods, which is ideally what we would want to see. We also see proportional yields between the 25 ml LB cultures and the 50 ml LB cultures, showing that the protocol, scales up nicely. So now I'd like to focus on a fully automated solution. So this is a workflow starting from cell pellets all the way through to purified DNA. And this would be better suited for those mid to high throughput users. This workflow uses the same kit, our MagBind endo-free MIDI kit, on dynamic devices links instrument. And the unique aspect of this system is that it uses multiple tools, a 24 pipetting head and a 24 magnet head to basically perform all the steps of the MIDI prep protocol. So essentially it is a liquid handler and a magnetic bead processor all in one instrument. You can also have different configurations here. So like a one-arm system that can actually interchange between the different tools mid-extraction run, or you can have a multiple arm system, which the tools you could use simultaneously. And I'll go into some of the advantages of that here in a second. But just to give you an idea of how this works, I do have a couple of videos. All right. So this first video shows the instrument swapping out the different tools. So currently it has the 24 pipetting head on. So after it's done with all the pipetting steps, we can drop that into its park position. And then we can go over and we can pick up the magnet head, which we'll use for the bind, wash, and elution steps. Skip ahead. So in the second video, we show how the instrument mixes our magnetic beads. So here, the magnet head, the magnetic rods are outside or above the tip covers, and it's going down as first as we can also do a couple of different types of mixing. So first it does a bottom mix to resuspend the beads, and then it could do a longer mix to really get the beads fully resuspended into each well. And so after you're done mixing, the magnetic rods will come down, will lower down into the tip covers. And this is how we will magnetize or collect our magnetic beads. And then once we have all of the magnetic beads collected, then you can transport them to the next plate, which would contain a wash buffer. Also shown here is our Easy Select 24 well depot plates. So these are a high capacity depot plate. So in each well, we can hold a total volume of 25 ml, or we have a working volume of around 16 ml with this magnetic head. So basically, this increased volume allows us to not have to split samples across different wells, or not have to split samples across multiple plates, which is what you would have to do in other magnetic bead systems. So going back to the workflow details, this system can process 24 samples in approximately 80 minutes, so it's very fast. That's one of the benefits of the magnetic bead processors. When we're talking about these large volume purifications, it's much easier to actually move the beads around than it is to pipette and transfer liquids. So that's why this workflow is much faster than some other solutions out there. And then this is also designed for cultures up to 50 ml. So if you needed something that was more high throughput, you could actually use a multiple arm link system where you would have the 24 head and the magnet head work simultaneously. So the 24 head would perform all the upfront steps. So again, the alkaline lysis, the neutralization, and the lysate clearance steps. And then the magnet head would perform the bind, wash, elute steps. So basically, you would start your first plate, and then after your 24 pipetting head was finished with the upfront steps, then it could actually start on the same steps for sample plate 2. And so if you took this staggered approach, you could actually process 96 mini preps in approximately 3 hours and 20 minutes. And then if you wanted to extend this further, if you wanted to run, you could run this continuously, and basically every 40 minutes you would produce 24 mini prep samples. So again, just to briefly review some of the data generated here. Here we compare the yields and purity of 3 different culture types, and the automated samples are comparable to the manual samples. We also might see a little bit less variation in the yields, which is ideally what you would want to see with automation. And then the purity ratios are right in line of what we would want to see as well. So I do want to also highlight some of our pre-scripted workflows for our Plasmid mini preps. And we have more options available, but I just want to touch on 3 here today. So these all use our new MagBind endo-free mini kit. First off, we have a solution for our MagBinder Fit 24, and this is similar to the MIDI workflow, and that is a semi-automated solution where those upfront steps are performed manually, and then the bind, wash, elute is performed on the instrument. This can process 24 samples per run. The manual processing time, again, it varies, but it's roughly around 30 minutes, and the automated processing time is 65 minutes. If you needed a mid-throughput solution, we have a workflow developed on the OpenChance Flex instrument with a 96 head. This is a semi-automated solution in that there is one pause step, and this is simply for reloading tips and reagents. The robot itself will actually perform all those upfront steps as well as the bind, wash, elute. So it's different in that regard from the MagBinder Fit workflow, but there is that one pause step for reloading reagents and tips. And then if you needed a higher throughput solution, we have a workflow on the Hamilton Microlab Star. So this is fully automated, and this can process 96 to 384 samples per run, or 1 to 4 sample plates. And the automated processing time is from 95 to 140 minutes, depending on how many sample plates you're running. All right, again, just some brief comparison data to review. So here we process 1.5 ml cultures, and we are achieving some high yields exactly in the range that we would want to see. And then again, we're still seeing very low endotoxin levels that would be endo-free grade. And similarly to the MIDI prep workflow, we are seeing a higher transfection efficiency due to the lower endotoxin levels. And here we have, we just tested the MagBinder Fit and the mini kit on across different culture media and plasmid types. And again, we're achieving very high yields exactly in the range that we would expect. And we are also getting the purity ratios that we would expect, indicating that we're getting pure DNA. So here's a quick snip of the data from the Hamilton workflow. These are from 1 ml culture, so we're getting very good yields here. Again, in between 7 and 8 micrograms, which is what we would expect. And again, the 260, 280, 260, 230 ratios are in the range that we would expect as well. All right, so this is just a quick summary of all the methods that we've developed and tested using our new mag-bind endo-free plasmid kits. With that being said, if you are working with a different system or if you have a unique application, we do have custom automated solutions available to you where we can work with you. We can have an initial consultation. We'll sit down with you. We'll figure out your exact needs, and then we can develop a custom workflow specific to what you're trying to do. We can also assist with the evaluation of our products, whether that be you can send us samples to our lab, we'll process them on the instruments that we would recommend, and then we can send you the DNA back for analysis so that you can ensure that it's suitable for your specific application. Or if you would like, just to test it manually, we can arrange that just a sample kit for you so that you can test it in your own hands. We also help with the implementation phase. So we'll go on site, we will install the workflow, we would train you and your team on how to operate and run the system. And so that is all I have. Thank you very much, and I will hand it over to Claire, and I believe we will field some of your questions.
Thank you, Jeff. Like he mentioned, we will be getting to some of your questions now. So if you have any questions about anything you've seen here today, please feel free to leave them below. But in the meantime, I do believe we already have a few in. So first up, do the lysate clearance beads work with magnetic processors?
Yeah, so excuse me. Yeah, I could take that one. So the lysate clearance beads are not designed for use with magnetic processors. If you're using our mag binder or a similar alternative, we would recommend either the lysate clearance syringes or centrifugation for the lysate clearance step.
Awesome. Okay, let's see. Next up, how long does it take to run 24 MIDI-sized samples?
So it really would just depend on how you're processing your samples. So if you're either automating or if you're processing manually, again, and if you are automating, it depends on which system you're using. So again, if you're using this, like the dynamic devices system that we referenced earlier, you could process 24 samples in approximately 80 minutes. If you were using a workflow with our mag binder, you could process 24 samples in roughly an hour and 40 minutes. Although with that, it is a semi-automated workflow. So there might be some variation from user to user with that.
Awesome. Thanks, Jeff. What types of growth media are compatible with this system?
I can take that question, Claire. So we have actually shown a lot of data with different growth media. One of the key features with both our kits is versatility. It can handle different culture media and different plasmid types. We have data with LB medium, terrific broth, 2XYT, as well as Plasmid Plus. And if you're working with any culture media that we've not tested, we would love to work with you. So do contact us and we can provide you recommendations and how to proceed with the culture media that you're using.
Great. Thanks, Karen. Let's see. Next up from our questions. Can this system be scaled up beyond MIDI size?
I can take that question as well. One of the advantages of using magnetic beads is scalability. And we do have preliminary data up to 100 mil. So again, if your application involves more than 50 mil of culture medium, do contact our scientists at Omega Bio-tek, and we will work with you in automating your unique experimental need.
And it looks like we have one more question open. How does this performance compare to, say, a spin column based kit?
I can take that as well. So the yields are very comparable when you compare spin columns versus magnetic beads. However, both of them work in different realms. In the sense, spin columns are more designed for a low throughput, more hands-on. But if you're looking for an automated approach, we would recommend using magnetic beads. Again, with magnetic beads, you have the added advantage of able to get high throughput. It satisfies the high throughput requirement as well as providing you with a more turnkey solution.
Thank you very much, Karen. It looks like that is all of the questions that we have for today. I want to thank everyone very much for joining us. If you have any other questions about anything you've seen here today, feel free to reach out to us at info@omegabiotech.com. Thank you, everyone.
Thank you.